Welcome to this lecture on RNA polymerases. Today, we're going to explore one of the most fascinating and essential molecular machines inside your cells. We will learn how your genetic code is read and copied, allowing your body to function and survive. By the end of this session, you will understand not just the biology, but also the math behind this incredible process. Let us dive in. Let us start by building an intuition for what this enzyme actually does. Imagine you have a priceless, ancient recipe book that you cannot ever take out of the library. If you want to bake a cake, you would not risk ruining the original book in your messy kitchen. Instead, you would carefully copy the specific recipe onto a piece of paper. In your body, your DNA is that priceless recipe book, safely locked inside the nucleus. RNA polymerase is the molecular machine that acts as the dedicated scribe, reading the DNA and making a temporary working copy called RNA. This ensures the original DNA remains safe while the cell gets the instructions it needs. Now that we understand the recipe book analogy, let us look at the formal biological definition. RNA polymerase is a complex enzyme responsible for synthesizing ribonucleic acid, or RNA, from a deoxyribonucleic acid, or DNA, template. This fundamental biological process of copying DNA into RNA is known as transcription. It is the very first step in gene expression. Interestingly, the complexity of this machine varies depending on the organism. In prokaryotes, like bacteria, a single type of RNA polymerase handles all transcription duties. However, eukaryotes, like humans, are more complex and have three main types, each specializing in producing different kinds of RNA. With the definition clear, let us explore exactly how this machine operates on the DNA track. The enzyme works by first binding to a specific starting region on the DNA called a promoter, which acts like a landing pad. Once securely attached, it unwinds the double helix to expose the genetic code hidden inside. It then reads the template DNA strand in the 3' to 5' direction, matching complementary RNA nucleotides to the exposed DNA bases. As it moves forward, it links these nucleotides together to build a growing RNA strand in the opposite 5' to 3' direction. This directional reading and writing is a universal rule in molecular biology. Understanding this mechanism leads us to a crucial realization about our health and survival. The importance of RNA polymerase cannot be overstated, as it is the absolute bottleneck for gene expression in all living organisms. Without it, your DNA is completely useless. In the real world, many life-saving antibiotics, such as rifampin, work specifically by targeting and jamming the RNA polymerase of bacteria. By stopping the bacterial scribe, the bacteria cannot make new proteins and quickly die. Conversely, certain deadly toxins, like the one found in the death cap mushroom, specifically inhibit human RNA polymerase, showing just how critical this enzyme is for our moment-to-moment survival. Before we move on to the mathematics of this process, let us visualize everything we just discussed. Here, you are looking at a transcription bubble. The large gray shape represents the RNA polymerase enzyme moving along the DNA. Notice how it has unwound the blue DNA double helix in the center? The bottom blue line is the template strand, which the enzyme is actively reading. The red line emerging from the complex is the growing RNA strand. You can clearly see the direction of movement indicated by the arrow and how the RNA is being synthesized by matching nucleotides inside this protected bubble. Now that we have a clear picture of the biology, Let us look at this process through the lens of speed and efficiency. Just like a car traveling down a highway, this enzyme moves along the DNA track at a measurable pace. We can calculate the length of the RNA molecule produced using a simple linear motion equation. The formula is L equals V times T. Here, L represents the total length of the newly synthesized RNA transcript, measured in nucleotides. The symbol V represents the velocity or speed of the RNA polymerase, typically measured in nucleotides per second. Finally, T represents the total time the enzyme has been actively working, measured in seconds. If the velocity increases, the enzyme builds a longer RNA transcript in the same amount of time. Let us apply this formula to a real biological scenario. Imagine a bacterial cell that needs to transcribe a gene that is exactly 4,500 nucleotides long. The bacterial RNA polymerase moves at a constant velocity of 50 nucleotides per second. We want to find out exactly how many seconds it will take to completely transcribe this gene. First, we rearrange our formula to solve for the unknown variable, time, giving us T equals L divided by V. Next, we substitute our known values, 4,500 divided by 50. The units of nucleotides cancel out, leaving us with exactly 90 seconds. This means it takes just 1.5 minutes for the bacterium to completely read the gene, highlighting the incredible efficiency of these molecular machines. That single enzyme was fast, but this brings us to an even more impressive phenomenon. In highly active genes, cells do not wait for one RNA polymerase to finish before starting another. If a cell needs a massive amount of a specific protein quickly, it will send multiple enzymes down the same DNA track, following each other in a convoy. This physical phenomenon explains how a cell can rapidly produce a massive amount of RNA from a single copy of a gene in a very short amount of time. It is a beautiful example of biological mass production happening right inside your cells. Let us see how we can calculate the size of these convoys. To figure out how many enzymes are in a convoy, we must break the problem into two manageable mathematical steps. The first step is to determine the total time it takes for one single RNA polymerase to travel from the beginning to the end of the gene. We use the rearranged length formula we saw earlier. T total equals L divided by V. Here, T total is the total travel time for one enzyme, measured in seconds. L is the total length of the gene in nucleotides, and V is the speed of the enzyme. By treating the entire gene as a single track and the enzyme as a train moving at a constant speed, we establish the time window during which new enzymes can attach. Let us put this first step into practice with a human gene. Suppose we have a human gene that is 24,000 nucleotides long. The human RNA polymerase travels at a speed of 40 nucleotides per second. We need to find the total travel time. We take our formula, T total equals L divided by V, and plug in our numbers. We divide 24,000 nucleotides by 40 nucleotides per second. The calculation tells us that it takes a single RNA polymerase exactly 600 seconds, or 10 minutes, to transcribe the entire gene. Now that we have this 10-minute window, we can move to the second step to find out how many enzymes join the convoy during this time. Now that we know the total travel time, we can determine the convoy size using our second formula. N equals T total divided by T interval. In this equation, N represents the total number of RNA polymerases actively transcribing the gene at any given moment. T total is the total transcription time we just calculated, measured in seconds. And T interval is the specific time delay between each new enzyme attaching to the start of the gene, also in seconds. This division tells us exactly how many enzymes managed to hop onto the DNA track before the very first one fell off at the end. It's a simple ratio that reveals the density of the transcription machinery. Let us finish our calculation for the human gene. We already found that the total travel time, T total, is 600 seconds. We are given that a new RNA polymerase attaches to the start of the gene every 8 seconds. This is our T interval. We want to find N, the total number of enzymes. We use our formula, N equals 600 divided by 8. The final answer is 75 RNA polymerase molecules. This means that at any given moment, there is a massive convoy of 75 molecular machines simultaneously reading the same gene, trailing one behind the other. This perfectly illustrates how our cells achieve rapid mass production of essential genetic messages. We have covered a lot of ground today, so let us summarize the key takeaways. First, RNA polymerase is the essential molecular scribe that copies the permanent DNA blueprint into a temporary RNA message. This process, called transcription, is the absolute foundation of gene expression and is critical for survival. Second, we learned that biology is not just about shapes. We can model this process mathematically using simple physics equations like L equals V times T. Finally, we saw that cells can achieve astonishing efficiency by sending massive convoys of these enzymes down a single gene. Understanding this enzyme gives you the key to understanding how the static code of life is actively transformed into the dynamic proteins that build our bodies. Great job following along.